Before we talk about Alzheimer's disease, plaques, tangles, or treatments, I want to start with a simpler question. Why do neurons, some of the most complex cells in the human body, lose the ability to maintain themselves? Alzheimer's is often described as a disease of toxic buildup. Amyloid plaques accumulate, tangles spread, neurons die. But that description skips something deeper. Healthy neurons are not passive. They constantly repair DNA. They regulate RNA splicing. They recycle proteins. They manage energy under extreme demand. Alzheimer's disease emerges when these maintenance systems fail. Today, I want to show you why NAD plus biology, especially its role in RNA repair, is becoming one of the most compelling resilience-based frameworks for understanding and treating Alzheimer's disease, not as a miracle cure, but as a way to restore the cell's ability to function. Alzheimer's disease is not rare. Today, an estimated 55 to 57 million people worldwide live with dementia. Between 60 and 70% of those cases are attributed to Alzheimer's disease. As populations age, this number is expected to rise dramatically. Despite decades of research, Alzheimer's remains incurable. That is not because scientists have failed. It is because Alzheimer's is not a single failure. It is a systems collapse. For many years, the amyloid hypothesis dominated Alzheimer's research. The idea was simple: remove amyloid plaques, stop the disease. Reality turned out to be more complex. Recent monoclonal antibodies, such as lecanemab and donanemab, finally demonstrated something important. Reducing amyloid can slow cognitive decline. This was a turning point. But the benefit is modest. It appears mainly in early symptomatic disease, and it requires careful MRI monitoring due to risks like ARIA. Amyloid removal matters, but it is not the full story. Attention has increasingly shifted to tau. Recent biomarker research shows that tau pathology begins long before visible tangles appear. Specific phosphorylation sites, serine 262 and serine 356, mark early, soluble tau assemblies. These early tau changes disrupt neuronal function before structural collapse occurs. This tells us something crucial. By the time neurons are visibly damaged, the disease has already been active for years. Amyloid immunotherapy is not without risk. ARIA, amyloid-related imaging abnormalities, can occur during treatment. The risk varies. It depends on the drug. It depends on genetics, especially APOE status. It depends on monitoring protocols. Large real-world registries now show that most ARIA cases are mild and manageable. But the need for monitoring reminds us of something important. These therapies intervene late in a long disease process. So we ask a different question. Instead of asking only what to remove, we ask what has failed. Neurons depend on energy homeostasis, protein recycling, DNA and RNA integrity, balanced immune signaling. When these systems fail together, pathology emerges. This is where NAD+ enters the story. NAD+ is often described as an energy molecule. That description is incomplete. NAD+ is a central currency of cellular maintenance. It powers mitochondrial metabolism. It enables DNA repair through PARP enzymes. It regulates stress responses through sirtuins. It shapes immune signaling. As we age, NAD+ levels decline, not only because synthesis drops, but because consumption rises. In 2025, a pivotal study revealed something unexpected. NAD+ depletion in Alzheimer's disease disrupts RNA splicing. Through a regulator called EVA1C, reduced NAD+ leads to systematic splicing errors. These are not random mistakes. They alter neuronal identity and function. When NAD+ levels were restored in disease models, RNA splicing fidelity improved. Neuronal function improved. Behavior improved. This was not plaque removal. It was functional restoration. This effect was observed across multiple layers. At the molecular level, RNA processing normalized. At the cellular level, neurons regained functional stability. At the system level, cognitive performance improved in models. Importantly, EVA1C dysregulation is also observed in human Alzheimer's tissue. This bridges model systems to human relevance. At this point, an important clarification is necessary. Human trials of NAD plus precursors like NR and NMN show clear biochemical effects in blood, but cognitive benefits in humans remain limited and inconsistent. This tells us something critical. Raising NAD plus systemically is not enough. The brain is selective. Mechanism matters. Target engagement matters. NAD plus is not only about supply, it is also about loss. Two major consumers increase with age and disease. CD38, an immune-linked NADase. PARP enzymes, activated by chronic DNA damage. If these drains remain open, replenishment alone will fail. This reframes Alzheimer's as a problem of metabolic leakage. The future of Alzheimer's treatment is not one drug. It is layered intervention. Amyloid removal reduces toxic burden. Tau modulation limits structural damage. NAD plus system repair restores cellular decision-making. Recent studies targeting CD38 in Alzheimer's models show improved metabolism, reduced inflammation, and restored cognition. This is resilience biology in action. Think of the system this way. NAD plus supply supports repair. NAD plus drains accelerate collapse. EVA1C links metabolism to RNA integrity. When balance is restored, neurons regain function, even in the presence of pathology. 3. Alzheimer's disease is not simply toxic accumulation. It is the loss of the cell's ability to maintain itself. NAD plus biology connects metabolism, immune control, and RNA integrity into a single framework. The future lies in earlier intervention, mechanism-driven trials, combination resilience therapies. The invitation is clear. Move beyond single targets. Design systems-level medicine. Measure repair, not just removal. That is how we turn Alzheimer's research into Alzheimer's resilience.